afternoon. Good afternoon. I wish I could ask you to remove your masks so that I can see your smiles, but by faith, I know you are smiling behind the mask. All right? So tutaendelea. Poleni sana, kumekuwa na foleni kubwa pale watu wakila chakula. I don't think the hotel had anticipated that. So tomorrow we're going to make it better. You have my word. Kuna wengine bado wako bado unakula, so watatupata tukiendelea. So for this session, I'm going to invite, we are going to first talk about the performance of the African eggplant. Eggplant ni nini kwa Kiswahili? Nyanya chungu. Mwaitaje wewe?
Nyanya chungu.
Nyanya chungu. Hey. Biriganya. Biriganya. All right, tutakuwa tukiongelea mambo ya biriganya. So performance of the African eggplant, entries across environments and hits for selection environment in northern Tanzania. Let me have your attention. May I have your attention, please? So we are going to have Dr. Fekadu Dinssa from the World Vegetable Center from Tanzania, who will be taking us through that presentation. Mpigeni makofi. Daktari.
Good afternoon, everybody. Yeah, my presentation is on performance of African eggplant, Solanum aethiopicum, entries across environment and hints for selection environment in northern Tanzania. There are a number of people who co-authored the breeding staff. And one of our senior researcher also, Dr. Peter Hansen from Taiwan. I've finished the presentation. Yeah, this one. African eggplant is one of African traditional vegetables. It is one of the two African traditional vegetables selected for as a strategic crop for breeding at World Vegetable Center. The crop is consumed as fruit vegetable and as leafy vegetable. I hope everybody knows this. There are three common species of African eggplant. Solanum anguvi, Solanum microcarpon, Solanum aethiopicum. Solanum aethiopicum is a very large species with some groups under it. Some of them are like gilo or fruit type. The sham group is leafy type, and the other one as a leaf and a fruit vegetable. And there is one group under this species, which is not commonly consumed, but known for its resistance to abiotic stresses, like, for instance, insect. Solanum aethiopicum, the Gilo group, is our focus breeding program in the current one. It has diverse morphological type of fruits at maturity, physiological maturity, again at marketable maturity. Marketable is when we harvest for cooking. Physiological maturity is when we keep until it gets the seed is mature for seed production. I think I pushed it 2 times. Yeah, research and development on African eggplant, like in any of the traditional vegetables, is very less. Both public and the private sector are not focusing that much, but currently getting momentum anyway. Farmers grow land races with few improved varieties available. There are 2 popular varieties, open pollinated varieties, DB3 and Tangeru White, originally released in Tanzania, but grown in East Africa and South Africa. In many cases, seeds are exported from Tanzania. The same variety, which is DB3, is recently released in Cameroon in 2020. Local and regional seed companies, we categorize seed companies into local, regional, and also multinational companies. The local and regional ones are marketing seeds of public varieties, varieties released by National Agriculture Research Systems. Multinational breeding companies develop hybrids, especially Rex1 and East-West. They have released some hybrid varieties from World Vegetable Center breeding lines. World Veg is developing improved lines for both public and the private sector. The private sector, the public sector, they use in their breeding program or directly evaluate and release in their target environment for commercial production. Yeah, please. Knowledge of genotype, environment, and the genotype by environment interaction is very important to determine the adaptation range of varieties, to identify a number of test locations, and the type of environment is required in a breeding program. Otherwise, we may over-test, or we may evaluate in few locations where other representative locations are out of picture. Such information is not available for breeders currently, and it is not, African eggplant is not exceptional. Results of multi-location trial on African eggplant is presented in this particular presentation. The study determined contribution of genotype, environment, and the genotype by environment interaction.
Kenya bora mngana nini? Mama anakufuatilia sana. Kufuatilia sana. Ni wakuloka.
21 genotypes were
you give her just now or you hear right away
in 7 environments.
Yani,
2 locations in 2017.
But I wish you could put on speaker, you hear.
2018. We consider here each location, each year, independent environment. So it adds up to 7 total environments. The locations vary in altitude, ranging from 765 to 1,500, let's say about. They also vary in soil characteristics, temperature conditions. The materials were evaluated in a randomized complete block design in 3 replications. Data collected, huge number of data collected, but for this particular presentation, fruit yield, wilt disease, and the farmer's preference rating. I hope many people want us to hear what farmers are doing. 65 female farmers were participated, and 58 male farmers participated in the participatory variety selection. Data analysis, both individual and the combined analysis of variants were conducted. The additive, main effect, and the multiplicative interaction analysis was carried out across the 7 environments. The model used has been given. Genotype by environment interaction by plot analysis was conducted to identify target mega environments and the winner genotypes in different locations or environments. Yeah, this particular table shows fruit yield and farmer's selection score and the wilt incidence. The red ones, if you see the red font lines, they are the winners across locations. And very interestingly, they are the ones also is less susceptible to wilt disease. And they are also receiving high rank on the scale of 0 to 4. 4 the best, 0 is the poorest. They are giving also farmers rank. Both female and the male farmers rank was high. And the data for yield was presented in by location, by environment. Then you could see the all the statistical analysis were highly significant, with the exception of one environment. This is combined analysis of variance using the 7 environments. You could see here the genotype difference among genotypes highly significant, environment highly significant, genotype by environment interaction is also highly significant. When we apportion the proportion of sum of squares of treatment, genotype by environment interaction received the highest, and environment contributed the second, with genotype the last. This is the AMMI analysis, the additive main effect and the multiplicative interaction analysis, where the genotype by environment interaction was broken down to 3 interaction principal components. These are significant interaction components. In principle, what are these principal components? They are standing for certain environmental factors. For instance, it could be temperature that is contributing the highest to the interaction, or it could be the moisture status, it could be soil fertility. We have not investigated on this, but this indicates the first interaction principal component contributes like 75% of the total sum of squares of the genotype by environment interaction. And the last is still significant, is contributing 6%. The AMMI analysis selected, very interesting in this, selected 4 highest ranking genotypes in each of the 7 environments. The first one, if you see, this one, 1, 2, 3, 4. These are the best genotypes according to the AMMI analysis that were identified for at Chekereni, which is in Moshi. And you can see some of the genotypes Let's see the green one. The green font, it is best in one of the best four in at least 4 environments. This shows a kind of stability, and the yield table also tells us this, this information. And this variety, this line is at least in 3 environments. And this one, at least in 3, the blue one, at least in 3 environments. This indicates these have very good potential for as a variety to be released. Yeah, this one is the G+E, the G+E biplot. It categorizes environments into a cluster of groups, similar environments into one, and also similar genotypes into one place. You can see the locations. This group is 1, this group is 2. Mainly 2 main cluster groups of environment. We have evaluated 5 environments, but everything is similar, so we could only test in 2 environments other than wasting more resources than by growing in 5 environments. This is very important for breeders. This kind of experiment should be done whenever resources are available in a more number of locations, more number of agro-ecological conditions. Then the indication on this, the indication is, 1, as a conclusion, significant roles of environment and the genotype by environment interactions were apparent from the highest sum of squares of treatment explained by environment and the genotype by environment interaction. AMMI selection identified 4 top entries at each environment. At least 4 entries tended to be stable across environments. The top 4 entries across environment were also highly tolerant to the wilt disease. This wilt disease is a complex wilt disease, which we are still investigating, bacterial wilt and fusarium wilt. Both exist in these farms. So maybe bacteria sometimes as a beneficiary there, but when you analyze it, you could find in the soil whether it is a pathogen or the beneficiary one. We have to sort out. But the wilt complex is fusarium wilt and bacterial wilt. The G+E biplot analysis suggested 2 different environments. From the 7 environments, identified 2 different ones. The current 5 trial locations could be reduced to 2 locations. Now it is much safer and very interesting for breeders who suffer from resource scarcity. World Veg Arusha station could serve as initial selection site. It tended to cluster in both locations, in both cluster groups. AMMI analysis partitioned genotype by environment interaction sum of square into 3 significant interaction principal component axes. But what we did not do here is, what are the environmental factors that is responsible for the principal component axis 1, axis 2, axis 3? Is it temperature? Is it soil? Is it moisture? Or any other a factor like soil fertility, soil characteristics? Some may be clay, the other one is sandy. So this we have to identify. This one we did in amaranth, and a very similar study. Altitude played role, temperature, and soil characteristics played major role in amaranth environment clustering. I'm worried, you know, I pressed then. Yeah, the last one is yield was negative. I did not put in this paper, but from behind, yield has negatively correlated with wilt incidence. Both female and the male farmers selected against wilted plots, let's say, wilted entries or genotypes. Both female and the male farmers selection was correlated with high yield. Female farmers and the male farmers selection scores positively correlated. The two also agreed. So probably the selection was conducted by gender disaggregated participatory selection. Now, if the husband and wife are there, if one of them go and select, still they represent the other. It has implication in resource, in time of farmers. So, but very interestingly, like the correlation of female farmers with wilt, the coefficient is a bit lesser than the coefficient that the male farmers correlated to wilt disease. So it is apparent that the wilt is becoming very, very important in Ngogwe, in African eggplant, and it must be an important breeding objective in the future variety development. Thank you very much.
Begin makofi tena. Before I let you go, is someone who has a question? Before I let you go? Yes, there's a question here. You have a question? One question, just one question. Quick one. Quick one, just one quick one.
Oh, yes, thank you. Number one, compliments to the presenter. Thank you very much. But I would like to ask, what are these environmental factors that you think influenced performance of amaranth against the genetic factors?
Yeah, like I was mentioning, in amaranth, it was as a reference. We did similar study in amaranth. The paper has been published in American Horticultural Technology Journal. The factors were altitude, temperature, and the soil characteristics. In that study, we included, we went as low as 20 meters above sea level, Dar es Salaam. We did with Mekocheni Agriculture Research Institute of Tari. So they have conducted the trial in the low altitude area, the coastal area, and it was very interesting. The soil was very important also because the sandy soil of the coastal area compared with a bit cooler season and a more volcanic ash of Arusha soil, and the more clay also. So those altitude, soil characteristics, and temperature played great role. But in Ngogwe, in this African eggplant, we did not conduct that kind of correlation. The number of locations we have is a bit low, but we are organizing how to go about and identify. But we know temperature played a role, because Moshi was, in Amaranth study, Moshi was clustered with Dar es Salaam, even though the physical distance is very near to Arusha. It is much hotter than here, this place.